Okay, so we can start now. First, can I just check that everyone can hear me? The thing is that I can't see the chat most of the time. So could someone just unmute themselves and tell me if they can hear me? Okay, so I do see the chat right now. They're saying we can. And you were able to see my screen before, right? Like, for example, right now, you can see my screen. Yes, okay, awesome. I just wanted to double check before I started. Okay, so again, I can't see the chat as I'm actually doing this, so it would be better if you guys could unmute yourselves, but in case you can't, because I know that mic can be a problem, I have certain checkpoints, so I'll just go back to the chat during those times and see if there's any questions in the chat. But if you want to unmute yourselves while I'm explaining, that's totally fine too. Okay, so let's get started. Hi guys, I'm Hania Nizami. I'm going to be explaining the urine concentration portion of this lecture. So I'm not going to be doing the whole thing. I believe the second half of this is going to be done by a different tutor, but up until around slide 14, I'm going to be explaining. And just so you guys have an idea of how I usually conduct these, I usually explain things either using a diagram or a drawing. And then in the next slide, I quiz people. So I cover up certain words and I want you guys to answer them. So in this case, I think a lot of the questions are actually at the bottom, but anyways, you guys can just help me answer them together. Okay, so let's start. I know this is not a good drawing, but I just wanted to start off with normal renal anatomy as we go forward. So right over here, you can see my afferent arteriole over here, and then it's going to form my glomerulus, which is pretty much a capillary network, right? And then it's going to leave and it's going to form my efferent arteriole, right? As you can see with my laser pointer. And then my efferent arteriole is going to form peritubular capillaries. And those peritubular capillaries play a really, really important role. So the reason that I'm bringing this up is that I want you to know that the role of peritubular capillaries is responsible for reabsorption. So what I mean by that is that anything in the fluid over here, anything in this yellow part is eventually going to leave and it's going to exit our body. So what I'm saying is that anything in the tubule, whether it be in the proximal, loops of Henle, distal, is eventually going to leave my body. The problem is that sometimes we don't want everything in this tubule to leave our body. Sometimes we want some of the substances in our tubule to go back into our body. And the way that we do that is that substances from the tubule are going to be reabsorbed and they're going to enter peritubular capillaries. And as you can see, these really straight peritubular capillaries are called vasa recta, and recta actually means straight. So I just want you to be familiar with these terminology. Let's quickly go over the anatomy of the tubules as well. So this is the Bowman's capsule right over here. It connects the glomeruli. As the glomerulus filters plasma, it's going to enter the Bowman's capsule. Then it's going to enter the proximal convoluted tubule, which is right over here. It's going to go down the descending loop of Henle, and then it's going to go up the ascending loop of Henle. It's going to enter the distal convoluted tubule, and this last part over here is called our collecting duct. So I just wanted to really quickly go over the anatomy so that we're all familiar with that. And then the other thing that I wanted you to know about is that this entire segment over here that we're looking at is called a nephron, right? And something that I want you to be aware of is that the top part of the nephron is located in the cortex. This middle part is called the outer medulla. And then this part right over here is located in the inner medulla. So I just want you guys to be oriented that the outermost part, everything over here is the cortex. And as we go more inwards, we enter the medulla, or specifically this segment over here, which is the inner medulla. The reason I want you guys to be aware of that is that I want you to start paying attention to what structures are in the inner medulla, because we're going to be going over that later. Okay, there is one other thing that I want to go over, which is that you see this segment over here. we said that this was my ascending loop of Henle, right? The thing that I want you to know about this segment right over here is that it is permeable to solutes. It's not permeable to water. It's only permeable to solutes, okay? And this segment right over here that I'm highlighting, the ascending loop of Henle, is actually responsible for secreting the solutes into the interstitium. And we're going to see this is so important because it's responsible for creating a concentration gradient in the interstitium and a concentration gradient of solutes. So I just want you to be aware about that. This segment right over here, the ascending loop of Henle, is permeable to solutes and is responsible for creating a concentration gradient in this segment over here, which is called the interstitium, and it's really important and plays a really key role. So we're going to cover that. Okay, like I said, I usually explain things with drawings, and then I quiz you guys in the next segment. So for the first question, so describe the mechanism by the blank of the loop of Henle produces the high medullary interstitial osmolarity. So what was the part of the nephron that I just talked about that's responsible for producing a high medullary interstitial osmolarity? In other words, a high medullary solute concentration. If you guys want to answer in the chat. Ascending loop, okay, good. If you guys unmute, that would be easier for me, but yeah, correct. It's the ascending loop of Henle. Okay, and then the second one is that describe the interrelationship between the loop of Henle, the collecting duct, so the tubules, and the blank that allows dilute or concentrated urine to reproduce. So pretty much what we're talking about is what reabsorbs the solutes, like from the tubule. Does anyone want to answer? What's responsible for reabsorbing the solutes? No, reabsorption. Somebody said. No, it's not proximal. Okay, I'll go over it again. So reabsorption, by definition, means that it's going back into the bloodstream. Okay, so if I say reabsorption, anything that's part of the tubule is automatically crossed out. Reabsorption means part of the bloodstream. And so the answer I was looking for was the vasa recta. So pretty much what this sentence over here is saying is that the fluid over here is going to get reabsorbed back into the vasa recta. And because what we mean by reabsorbed means back into our bloodstream, and it means it's going to be staying inside of the body. Is that clear? I just want to make sure before I move on. Okay, people are saying yes. Okay, so let's talk about, so remember I just talked about that it's really, really important that right over here in the loop of Henle, I create what's called a concentration gradient. Specifically, I want a very high concentration of solutes over here, deep in the inner medulla, and I want a lower concentration closer to the cortex. So I want to have an increasing concentration gradient as I move down. But why is that so important? Pretty much the reason that it's so important is because the job of the kidney is to excrete certain substances, right? There are certain substances in our body that if they stay in our body, they're going to become toxic. So the role of the kidney is to excrete them. The problem is if there was no concentration gradient and we just reabsorb them normally, I would need a lot of water. I would need 4 liters of water. And so what a concentration allows us to do is to selectively sort of pick only the substrates to be absorbed, for example, and it pretty much just decreases the amount of water that's needed to excrete certain substances. So I'm just going to repeat that again. The reason we need a concentration gradient is because it would require way too much water to excrete substances. And so the way that we can combat that is if we create an increasing concentration gradient, we can selectively reabsorb only water, and therefore we're excreting substances more easily. So the main idea just of this whole thing, in case you didn't understand anything I just said, is that we need to create a concentration gradient in the loop of Henle, and that's going to allow us to save water and not use as much water to excrete substances. Okay, so before I go on, I just want to review the concept of osmosis, because the concept of osmosis is so relevant to the kidneys, and it's so important to understand this before you understand the kidneys. So as you can see, I have a compartment on the left side of my screen, okay, and I have a low solute concentration. The S's are solutes, and you can see there's very few solutes on the left side of the screen. On the right side of my compartment, or the right compartment I have on the screen, I have a very high solute concentration. You can see a lot of S's over here, which means a very high solute concentration. And they're separated by this dashed line in the middle, which represents a semi-permeable membrane. So what I want you to know about osmosis is that osmosis is the passive movement of water across a semi-permeable membrane from a low solute concentration to a high solute concentration. So that is so important. That definition you should know by heart. And the main thing that you should know about that is that water is going to go from a low solute concentration to a high solute concentration. And this is just going to be really important in terms of understanding the kidneys. Okay, water moves from a low solute concentration to a high solute concentration. That is the main thing that I want to emphasize to you guys. So let's take an example, and let's pretend that this left side is the tubule, and then the right side is the interstitium. Okay, in the context of this, if I have let's say that I decrease my solute concentration, do you guys think that there's going to be more water going from a tubule to the interstitium, or less water going from the tubule to the interstitium if I decrease my solute concentration? Okay, good. I'm seeing some answers, and they're saying more. Exactly. So you guys understand the concept that means. Because what I'm trying to say is that, like I said, while water moves from low solute to a low solute concentration to a high solute concentration, and the main thing to also know is that the more of a difference there is, the more water that's going to move out. So that's good. You guys understand the concept. The next thing that I want to go over is this loop of Henle over here. So as you can see, I have my descending loop, and then I have my ascending loop over here. And there's a couple things I want you to know. The first thing that I want you to know is that the descending limb is only permeable to water. That's what I drew over here. So the descending limb is only permeable to water. Okay, that's the first thing you need to know. The second thing you need to know is that the ascending limb is the opposite. The ascending limb is only permeable to solutes. Remember I said that in the beginning, the ascending limb is only permeable to solutes, okay? And the third thing that I want you to grasp from this diagram over here is that notice how the solute concentration goes from low to high. The solute concentration increases as you go down into the medulla. Okay, so those are the three things that I really want you to take away from this diagram over here. Okay, and then from that, we can kind of answer the questions over here. So the first one, building of a blank interstitium. So the interstitium in this diagram, is it hypertonic or is it hypotonic? In other words, is it concentrated in solutes or is it not concentrated in solutes? Okay, good. I see two answers for hyper. Exactly, it's hypertonic. I think somebody said hypo. So let me just explain it again. You see how there's a high concentration of solutes, especially the deeper that we go. This means that the interstitium is hypertonic in compared to the tubules. Okay. So that's the first one. And then the second one is as we go down the loop of Henle, is the concentration of solutes increasing or is it decreasing? Is the concentration of solutes increasing or decreasing as we go down the loop of Henle? Increasing, good. So that's what's meant by this phrase over here, concentrating by a countercurrent multiplier. So basically what they're telling you is that the first step is that first we have to make the outside interstitium, this area right here, we have to make it hypertonic. Okay, we have to make it really concentrated in solutes. And then the second step that we're going to go into is we have to make sure there's a gradient. We have to make sure that the bottom is very high in solutes and then the top is comparatively lower concentration in solutes. So two things we have to do, we have to make the interstitium hypertonic, and the second thing we have to do is we have to make a concentration gradient that increases as we go down. Okay, good. So now I'm going to be explaining what I just said in words using this diagram. Okay, so over here, you can see I have my loop of Henle, right? And right now my loop of Henle, everything is the same concentration. Everything is 300 milliosmoles. The reason for that is that the fluid that's coming in from the loop of Henle is actually coming from my proximal convoluted tubule. Remember, there's my proximal convoluted tubule, and then it goes down and it starts to enter my descending loop of Henle. The fluid in my proximal convoluted tubule is isotonic with plasma. In other words, it's 300 milliosmoles, which is the same concentration of solutes in my water. Okay, so the fluid that first enters in my tubule is isotonic with water. It's 300 milliosmoles. But remember, I don't want my fluid to be isotonic. I don't want all of this to be the same. I want a very high concentration in my interstitium. And the second thing that I want to do is I want to create a concentration gradient in my interstitium. So let's go over that. Let's go over cycle one. So just to be really, really clear and make sure everyone's on the same page, the goal of cycle one is to create a concentrated interstitium. The goal of cycle one is to create a concentrated interstitium, which is this area over here, okay? So the way that that's gonna happen is that if you look, I have my ascending loop of Henle, right? And I told you that my ascending loop of Henle is only permeable to solutes. Okay, it's not permeable to water, it's only permeable to solutes. So what my ascending loop of Henle is going to do is that it's going to start pumping out some solutes from the tubule into the interstitium. Okay, and it's specifically trying to reach this target goal of a difference of 200 milliosmoles, right? 400 minus 200 is 200. So if I look, just to repeat it again, the first step is I look at my ascending loop of Henle. It's permeable to only solutes. It wants to create a very concentrated interstitium. And the way that it's going to do that is that it's going to start pumping solutes from the tubule into the interstitium until it reaches a difference of 200 milliosmoles. Okay, that's the first step. The second step that's going to happen is that, remember, I told you my descending loop of Henle is only permeable to water, right? My descending loop of Henle is only permeable to water. So these pink arrows that you see are indicating water that is leaving from my tubules into my interstitium, right? And because water is leaving from my tubules into my interstitium, is the fluid over here going to get more concentrated or less concentrated? I'm going to ask that again. Is the tubule in my descending loop of Henle, in my descending limb, when water leaves, is it getting more concentrated or less concentrated? More, good. Right, so now I have a more concentrated fluid in my descending loop of Henle, right? The next step, now remember that fluid is constantly going in and out, it's constantly flowing through the tubules, right? So what's going to happen is that, remember how I told you that the proximal convoluted tubule has is isotonic with water and it has a concentration of 300 milliosmoles, that fluid is now going to enter into my descending loop of Henle. So that explains this 300 over here. This is the fluid that just entered into my descending loop of Henle from my proximal convoluted tubule, and that's why its concentration is 300, right? On the other hand, what's happening is, like I told you, fluid is constantly moving through the tubules. And so now this fluid over here that had a concentration of 400 has now moved over and is now in the ascending loop of Henle, right? The fluid that used to be in the descending limb has now moved over and is now in the ascending loop of Henle. So if you look at that, look at what that's creating. I have a low concentration up top over here, and I have a high concentration at the bottom over here, or rather I should be showing it over here. So I have a low concentration up top over here because this came from my PCT, my proximal convoluted tubule. I have a higher concentration over here because of the process that we just talked about. And so you can see it's the beginning of cycle 2, because the goal of cycle 2 is to create a concentration gradient. Okay, so what did I say the goal of cycle 1 was? The goal of cycle 1 was to just create a hypertonic interstitium. I wasn't worried about concentration gradients. Now in cycle 2, I do have a hypertonic interstitium, but I want to create a concentration gradient, okay? So like we talked about over here, you can see that all of this fluid over here was originally the fluid that was here, right? So this fluid right over here is the part that was all in the ascending loop of Henle. And then this segment right over here used to be in the descending loop of Henle, but now it's in the ascending loop of Henle. So what's going to happen is that it's essentially the same process. Remember I told you my ascending loop of Henle is only permeable to solutes. And I remember that I told you that the ascending loop of Henle, its goal is to create a difference of 200 milliosmoles. And so now it's going to start doing that. It's starting to create a difference of 200 milliosmoles. But because of what we talked about before, the interstitium at the top is less concentrated and the interstitium at the bottom is more concentrated. Okay, so the first step again is the same as cycle 1. We're just pumping out more solutes and we're trying to keep the difference of 200 milliosmoles between the tubular fluid and the interstitium. Okay. The second part, again, we're going through the same process again. This time, because I have a low solute concentration in my descending limb and a higher concentration in my ascending limb, think about what the definition of osmosis is. The definition of osmosis is that water is going to move from a low solute concentration in the descending limb to a higher concentration in the interstitium. So what that means is that water is going to passively flow from the descending limb into my interstitium. And because of that, because water is flowing out of my descending limb, this fluid right over here is going to get more concentrated, right? Fluid in my descending limb is getting more concentrated because water is leaving. And then you can see it ends pretty much the same way. However, this time when the fluid moves, it's getting more and more concentrated, right? This value used to be 400 in cycle 1, and now it's 500 in cycle 2. And so what you're seeing is the concentration or the concentration gradient is just increasing. The more cycles that we go, the more and more pronounced the concentration gradient is going to become. And you can see that over here. If you keep doing more and more cycles, you're going to get a very pronounced concentration gradient where the bottom of the loop of Henle is like about 4 times that of the top of loop of Henle. Okay, so I hope everyone understood. I'm going to be going through the text at the bottom here, and we're going to answer it together. Okay, so in the beginning, we've already talked about this. The proximal convoluted tubule has an osmolarity of 300 milliosmoles. And then from the proximal tubule, the fluid is going to enter into the descending loop of Henle. And remember, our goal is to create a hyperosmolar interstitium in cycle 1, and then we also want to create a concentration gradient in cycle 2. Okay. So the first question is, at what part of the loop of Henle, or at what part is salt being pumped out of the tubule? What was the name of that segment where salt is being pumped out of the tubule? Okay, somebody said, okay, ascending limb, good. And somebody asked, do we need to know the numbers? Honestly, I'm not sure. I would be familiar with the major ones. I would at least know that the proximal convoluted tubule has 300 milliosmoles. But I think a lot of the other numbers aren't as important. I don't think it's as important as understanding the concept. Exactly. That's what I can see is that the concept is more important. Okay, and everyone was right. The ascending loop, or the ascending limb of Henle, I guess I should say. But yeah, the ascending limb. Okay, and then I told you that water cannot follow the solutes because the ascending limb of Henle is impermeable to water. It's only permeable to solutes. Okay, and because the salts or the solutes are being pumped out of the ascending loop of Henle, is this going to increase or decrease the osmolality of the interstitial fluid? Okay, so I'm just going to repeat that. Because I'm pumping salts from the ascending limb of Henle, is this going to increase or decrease the osmolality of the interstitial fluid? Increase, good. Okay, and then what is it going to do to the tubular fluid? If salt is being pumped out of the tubular fluid, is that going to increase or decrease the osmolality of the tubular fluid? Decrease, exactly. So you guys are understanding the concept here. That's good. Okay. The second part is the increased osmolality in the interstitial fluid causes water to move from blank into interstitium until the osmolality of the descending tubular fluid equalizes. So okay, so the increased osmolality in the interstitium is going to cause water to move from where into the interstitium? Where is the water coming from into the interstitium? What part? Good, descending loop, because I told you that the descending loop is permeable to water, the ascending loop is impermeable to water. Good. Okay, you guys got that right. Okay, new fluid from where enters the tubule of the descending limb? Where did I tell you what part of the nephron does new fluid enter? Isotonic fluid specifically. The fluid that enters the descending limb, where is it coming from? Okay, I see one answer. Good, proximal tubule, exactly. And it's isotonic. Okay, then after a few cycles, a blank interstitium is created. Is it a hyperosmolar or hypoosmolar interstitium? Hyper, good. And then the second question is concentration or osmolality increases towards blank. So increases towards what part of the nephron? Remember I told you the nephron has an outer part and an inner part. What was the name? Increases towards the medulla, exactly. The inner part is called the medulla, and it has a high concentration of interstitium. Okay, then in equilibrium, the water and salt that are continually deported into the interstitial fluid are transported out of the medulla by blank. Good. Vasa recta. Exactly. Remember? So the key concept that you should be getting with that very last part is that as the solutes enter into the interstitium over here, they're eventually going to go into the vasa recta. And remember, the vasa recta are blood vessels. And that means that eventually, if things are entering the interstitium, they're going to go into the blood vessels and they're going to stay within the body. That's the main concept that that last part is trying to say. So again, I just really want to emphasize the solutes that stay in the tubules are going to be excreted out of the body. And the ones that go into the interstitium will eventually go into the vasa recta. The vasa recta goes back into the bloodstream and it stays within the body. Okay, so inside the body, outside the body is the bottom line. Okay, this is a checkpoint. Does anybody have any questions for me? Anything that they didn't understand? Again, you're more than welcome to unmute. Or if you want me to re-explain anything. Okay, somebody's saying all clear. Okay, all good, awesome. Okay, the next part that we're going to be going over is called the anti-diuretic hormone. In other words, called ADH. You guys are really lucky because ADH, or anti-diuretic hormone, tells you its function by the name itself. So we all know that anti means decreases. Anti means less, right? Or to stop. So anti decreases, and uretic means urine. And so anti-diuretic means it decreases urine. Now, if you think about that for a second, if I'm decreasing urine, that means that I'm decreasing the amount of water that's in urine, and that is going to lead to more concentrated urine. And so the bottom line over here is that the role of ADH is to concentrate urine by absorbing water. Okay, and this is going to lead to decreased urine output, which is in the name itself. Okay, so the role of ADH is to concentrate urine by absorbing water. This is like the main thing to understand about antidiuretic hormone. It acts by absorbing water in order to concentrate the urine. Okay, and that's going to decrease the urine output. Okay, so let's go in this diagram over here. You guys already understand this whole segment. Over here, we know this is the proximal convoluted tubule. It's isotonic with plasma. It gets more concentrated as it goes down. And then as it goes back up, it's pumping out solutes. And because it's pumping out solutes, it's going to become hypotonic. Okay, like we've already gone over this. As you go down the limb of Henle, you're losing water. It gets more and more concentrated. As you go up the ascending limb of Henle, you're losing solutes, and that's going to make it less concentrated. Okay, by the time it reaches this segment over here, which is called the distal convoluted tubule. Okay, so what I want you to know is that you can see ADH, right? ADH, the first thing I want you to notice is where is it acting? Because I see this a lot in MCQs. So where is ADH acting? It's acting on the collecting tubules and a little bit on the distal convoluted tubule. So ADH acts on the collecting tubule and a little bit on the late distal convoluted tubule, okay? But most of it is on the collecting tubule, okay? And what did we say the role of ADH is? We said that the role of ADH is to reabsorb water, right? Is to absorb water. So I'm removing water from the tubular fluid and I'm moving that water back into the peritubular capillaries, and that peritubular capillaries are going to go back into my bloodstream and they're going to stay within the body, okay? So think about that. And so because water is being removed from the tubular fluid, that means that the urine is getting more and more concentrated. You can see that the urine is getting more and more concentrated. Okay. So that's the main thing that I want you to know. I want you to know where it acts, primarily on the collecting duct. I want you to know its role is to absorb water or reabsorb water back into the bloodstream. And because it's reabsorbing water, that's going to make the urine more concentrated. Okay, those are the main, really important things to know. Let's see if you guys understood, if you can answer these parts. If ADH is present, water will follow the osmotic gradient and will move from blank into the blank until equal osmolality is achieved on both sides. So where is it moving? Where is the water moving from? Okay, it's moving from the tubular to interstitium. Perfect, guys. Okay. So as a result, the osmolality of the fluid approaches the osmolality of the interstitium fluid at the deepest part of blank. So think for a second, if I look at my diagram, what part of the diagram has the most concentrated tubular fluid? Or essentially what it's asking, like the concentration of this part resembles what part over here? How would you describe this portion? Medulla. Well, you guys have a lecture and you guys are just cheating or something, but you guys are right. So all they're saying is that the concentration at the end of the collecting duct is about the same as the medulla part over here, or the part that's sort of in between the two limbs of Henle. Okay. So now let's sort of inverse this and let's look at what happens if I don't have ADH, right? So if I don't have ADH, that means that I cannot reabsorb water, right? So all of the water is going to be staying inside of my collecting duct, or it's going to be staying inside of my tubule. And what that means is that my urine is going to be very, very, very dilute. Okay, so I'm going to be losing a lot of water, right? If I cannot reabsorb the water back into my bloodstream, if I don't have ADH. The other sort of secondary thing you want to pay attention to that I didn't really talk about over before is that we are absorbing water. We're mostly absorbing water in the collecting tubule, but we're also absorbing a little bit of solute. Okay, we're absorbing a little bit of solute. So not only is my collecting duct getting more dilute because it's keeping so much water, it's also losing a little bit of solute. And that's going to contribute to a decreasing concentration, and that's going to make my urine very, very dilute if I don't have ADH. That is the main concept over here. Okay, so the first question, the tubule is blank as it reaches the late distal tubule. Okay, so is it hyperosmolar or hypoosmolar as it reaches this part over here? Hypo, good. Okay, if ADH is not present, the luminal membrane of blank and blank remain impermeable to water. So what are the two parts that ADH acts on? The two parts of the tubules. Collecting duct and distal. Good. That's kind of right. And then the other thing that I told you about is that there is a small amount of solute reabsorption, and that's also going to contribute to very, very dilute urine. And that's going to make hypoosmotic urine, right? It's going to be less concentrated to the plasma than the plasma. Okay, so I have a practice question for you over here. I'll just read it out and then you guys can answer in the chat. So nephrologists at a research hospital are investigating the physiological changes that occur in diabetes insipidus. The group develops a technique that permits sampling of tubular urine in experimental animals with physiology similar to that of humans. The animals then undergo hypophysiectomy, after which tubular fluid samples are obtained from multiple sites throughout the nephron. In the absence of anti-diuretic hormone, fluid of which of the following sampling sites is most likely to have the highest osmolarity? So you can honestly ignore everything else, but that last sentence, absence of ADH, where is the tubular fluid going to be the most concentrated? A, B, C, or D? So A, B, C, or D, E, or F? Okay, so one answer. Does anybody else want to answer? Okay, the correct answer is C. I saw someone say F, but just keep in mind the key part of this thing is the absence of anti-diuretic hormone, right? And remember I told you if we don't have ADH, we cannot reabsorb the water, or sorry, the fluid that's over here. And because of that, the water is going to stay inside the tubule. And because it's staying inside the tubule, this part's actually going to be very dilute. Okay, but the question is asking what is the highest osmolarity of tubular fluid, right? So we're asking what part is the most concentrated. And this part right over here, C, is the most concentrated. Because remember, I'm losing water as I go down here, which makes this part very, very, very concentrated. And then I start to lose solutes, and that's going to make it less concentrated. So that means that the most concentrated part is right over here, as I've lost all of my water and I haven't started giving up my solutes yet. Does that make sense? Is that more clear why it's not F? Okay. If it said something like, you know, ADH was present and they were pointing specifically to somewhere like over here, and that was like G or something, then that could be a possible answer. But since ADH is not present, that means that the fluid is very dilute, and that means that this is the most concentrated. Okay, so over here, what they're just talking about is urea, okay? So the main thing that they're trying to communicate over here is urea. You can see I have my tubules, and they're just explaining to you guys how urea gets, it's kind of weird. It goes back and forth a lot. So it gets reabsorbed. Remember, reabsorbed means I'm going from my tubule into my bloodstream, and then it gets secreted, which is the opposite of reabsorption. It means it's going from my blood into my tubules, and then it's going to get reabsorbed again. So it's going like into the blood, into the blood, out of the blood, and then back into the blood. And then finally, it's going to be excreted. So urea is kind of like special like that. It's going to be reabsorbed in the proximal convoluted. It's going to be secreted, which means it goes from the blood into the tubules, and it's going to be reabsorbed again. So it's going back and forth between the blood and the tubules, essentially. And then they mentioned two transporters over here, or actually three transporters. I'm going to show you that in the next slide. Okay, so over here, this is my bloodstream, right? This is my bloodstream right over here on the right side of my screen. And this is my tubular fluid. So keep in mind, this is the urine over here. This is the part that's going to leave my body. And this is the part that's going to enter and stay inside my body, right? This right over here is what we call the basolateral side. And this side over here is what we call the apical side of the cells. Okay, and what they want you to know is that when urea goes from my blood into my tubular fluid, which is called secretion, sorry, when it goes from my blood into my tubular fluid, it's going to use the transporter UT2. In other words, urea transporter 2. Okay. On the other hand, if the urea is going from my urine or from my tubular fluid across the cell into my blood, it's going to first use urea transporter 1, and then it's going to use urea transporter 4, right? So urea transporter 1 is on the basolateral side. Okay, I would know the location of where these transporters are. So it's on the basolateral side, which connects the tubule to the cell. And then urea transporter 4 connects the cell to the blood. Okay, so this process is called reabsorption, where fluid goes from my urine back into my bloodstream to stay inside of my body. And this process is called secretion, where urea goes from my blood into my tubular fluid. Okay, I have like a silly little mnemonic over here. This is just like how I used to study. But the way that I would memorize this is that you could say UT2 goes to the loo. Loo is like the British way of saying bathroom. And what that will help me remember is that UT2 is part of secretion, right? UT2 is responsible of moving the urea into my urine, or in other words, into my tubular fluid. And that tells me that UT2 is responsible for removing the urea from my blood into my tubular fluid. So that's how I would remember that one. And then the second one is UT1 and 4 really love gore. So gore, or like gory movies, are usually like horror movies that have a lot of blood. And so that helps me remember that UT1 and UT4 are responsible for removing urea from my tubular fluid back into my blood because they love gore, right? So they're going to move back into my blood. Okay, so UT2 goes to the loo. That means it's going towards the urine for the tubular fluid. UT1 and UT4 love gore, and so they're going to go back into the blood. The last thing I just want to say is just pay extra attention to UT1 being on the basolateral side, that's this side, and UT4 being on the apical side of the cell, because again, sometimes they like to test on that. Okay, so what they're showing you over here, remember how we talked about how the nephron, the outer part of the nephron is called the cortex, the inner part of the nephron is called the medulla, and then if you go even inwards, it's inner medulla. What they're showing you over here is that the inner medulla is very, very permeable to urea. So the thickness of this is trying to show you how permeable the segment is to urea, right? And so if you have an understanding of the anatomy, all you have to remember is that the inner medulla is permeable to urea. And if you can remember which parts are in the inner medulla, like if you're able to draw it out, then you would know it's, you know, the end of the descending limb, the beginning of the ascending limb, and the collecting duct. So that's what they're trying to convey to you over here. So let's just quiz ourselves on the topic below. So the inner medullary blank and blank have a higher permeability to urea. So what are the two structures that you see on the slide that are very thick and are very permeable to urea? Yeah, the ascending and descending limb, and then what's this part right over here? Collecting duct, exactly. Yeah, so descending and ascending limb. This segment right over here is very permeable to urea. Okay, and then that also answers the last question, which is that the inner medullary collecting duct, this part right over here, is a major site of urea reabsorption. So that's just the last thing I want to talk about. So we talked about how all of these segments right over here at the bottom are permeable to urea, right? But the other thing that I want you to know is that the difference between these two is that this is responsible for secretion. The ascending and descending limb are responsible for secretion. So that means that the urea is going from blood into the tubular fluid versus the collecting duct, which is also permeable to urea, but this time it's the opposite, it's reabsorption. So it's going from the tubular fluid back into the bloodstream. That's called reabsorption. So ascending, descending limb do secretion, collecting duct does reabsorption of urea. Okay, so over here we have a graph, right? This is showing us the osmolality of tubular fluid. So again, high osmolality means very high concentration, low osmolality means very dilute. And if it's tubular fluid, you can think of it as urine, because eventually tubular fluid is eventually going to become urine. Okay, so what that means is that if I look at my y-axis over here, the higher the value, the more concentrated the urine is. And then if we look at the x-axis, this just means that we're going along the tubules. So I start with my proximal tubule, I go up and down the Henle loops, the distal tubule, collecting duct, and then I end with my urine. And so what you can notice is that this top line over here that I'm highlighting, this is the concentration of urine when we do have ADH. And then this bottom line that I'm highlighting over here is the concentration of urine when we don't have ADH, right? And so what you can notice is that the first thing I want you to notice is that the difference is most notable in the distal tubule and the collecting duct, right? And that should make sense to you, especially the collecting duct part. That should make sense to you because the effect of ADH is mostly on the collecting duct and then a little bit on the distal tubule as well. So the reason there's such a big difference over here is because these are the two parts that are most affected by ADH, right? The second thing I want you to notice is that you really have to understand that if I have ADH, if I have anti-diuretic hormone, I'm reabsorbing water back into my body. Because I'm taking the water outside of the fluid, this is outside the tubular fluid, this is going to make the tubular fluid concentration very high, right? It's going to make the urine very concentrated. That's the main concept of ADH. On the other end of things, if I don't have ADH, I'm not reabsorbing water from the tubular fluid. Because water is staying inside the tubular fluid, I'm going to have a very low osmolality, which means, in other words, I have very dilute urine. It's the main concept. Okay, so now let's try to apply our knowledge over here. So in water restriction, the kidneys maximally blank the urine. So are they going to concentrate the urine, or are they going to dilute the urine in water restriction? So my body is already low on water. Do you think that they're going to concentrate or dilute the urine? Concentrate, good. It's going to concentrate the urine, and that should make sense to you because if I concentrate the urine, I'm losing less water. And in cases of water restriction, my body's already low on water. So I want to be reabsorbing the water. And if I'm reabsorbing the water, that's going to concentrate the urine. Okay, and that's going to excrete a blank volume of water. Is that going to be a high volume of water or a low volume of water in cases of water restriction? So I'll ask again, in water restriction, is there going to be a high volume of water inside the tubules, inside the urine, or is there going to be a low volume of water in the urine? Low, right? Exactly. It's going to be minimal volume of water, because if water restriction, my body's low on water, I want to be keeping it inside the body. I don't want to be losing it through my urine. Okay, so on the other hand, ingestion of excess water, so this is a state that my body already has a lot of water, causes the kidney to produce a low or high volume of water. And is the urine going to be concentrated and dilute? So tell me the volume and tell me the concentration in cases of excess water. High volume, good. And is it going to be concentrated or dilute? Good, less concentrated. Large volume of water, that's very dilute, okay. Okay, so remember we talked about the descending loop of Henle, the ascending loop of Henle, and how it enters the distal convoluted tubule. At the end of that, at the part right before it enters the distal convoluted tubule, is that fluid hypertonic or hypotonic? Right before the fluid enters the distal convoluted tubule, is it hyper or hypotonic? Hypo, right? Because I just lost all of my solutes. Okay, so in cases of restricted water intake, right? So my body is low on water. Is this going to increase or decrease the levels of ADH? My body is low on water. Is it going to increase or decrease the levels of ADH? Increase, good. Okay, and as a result of that, the osmolality of the tubular fluid, is that going to increase or decrease? In the presence of ADH, is the osmolality of tubular fluid going to increase or decrease? It's going to be high, increase, exactly. Okay, the bottom two over here. On the other hand, without ADH, very little or no water will be absorbed in the collecting dust, and thus a blank urine, which is blank tonic to plasma, will be produced. So is it hyper or hypotonic? Hypotonic. Yes. And then it's also diluted, which is pretty much saying the same thing. So again, without ADH, I'm not reabsorbing the water staying in my tubular fluid. It's going to be hypotonic. It's going to be very less concentrated. Good, you guys understand the concept. Okay, so let's look over at this graph, see what's happening over here. And the x-axis, I have water, which pretty much this is telling me the percentage of water. You can think of it like as part of the tubular fluid. So if I have a very high percentage of water as my tubular fluid, that means that the tubular fluid is very dilute. And then if the concentration of water is very low, that means that the tubular fluid is very concentrated, right? So what they're showing you over here is that only 20% of water, or like whether water is going inside of my body or outside of my body, only 20% of that is controlled by ADH. Okay, most of the water reabsorption actually happens beforehand. And the way that I think of that is that we still want to be saving, no matter what the situation is, no matter even if I've had like a lot of water, a little water, I still want to be saving 80% of my water. Okay, the next of the 20% is under the control of ADH, and that's going to depend on, am I hydrated, and other factors, right? Am I sweating, blah, blah, blah. Okay, so what they want you to understand is that ADH actually only controls 20% of water reabsorption. It only has like a fairly small role compared to what you might expect. You know, most of it happens before the collecting duct. Okay, which I kind of spoiled the next one for you, but where are the two places that ADH acts? I know I asked this before, but I really want to make sure because it does come up on MCQs sometimes. Where are the two places that ADH acts? What part of the tubules? Collecting duct, good. And distal convoluted tubule, good. Okay. And then let's just try to apply our concepts over here. If we're looking at the graph, and I told you that the higher values means more dilute, right? Or like a higher water concentration. Do you think that this part over here is with the presence of ADH, or it's without the presence of ADH? If I have a lot of water inside of my tubules. Low, good. Right? If I have a lot of water that's still inside of my tubules, it means I'm not reabsorbing water, which means I don't have a lot of ADH. And then you can kind of guess that this line over here is in the presence of ADH. Because there's ADH, it's concentrating my tubular fluid, which means my water concentration is going down, and the water is going back into my bloodstream. Okay, so let's solve this part over here. If ADH is present, most of this fluid will be blank, and the urine will be blank. So most of the fluid will be what, and the urine will be what? Okay, I was looking for reabsorbed. Most of the fluid will be reabsorbed. Concentrated is also right. If you're looking at it from the perspective of the tubular fluid, that is true. If there's ADH, the tubular fluid is going to be more concentrated, which I believe is the second answer right over here. Yes. Okay, so over here, they're just telling you about renal blood flow and effective renal plasma flow. Remember that the difference between effective renal plasma flow and renal blood flow is that renal blood flow has hematocrit, which means that it includes RBCs, okay? Versus effective renal plasma flow doesn't include RBCs or the hematocrit, right? And then the concept that I think he's trying to explain to you over here is that the renal blood flow is very, very high, right? But if you look at my urine flow, it's much lower than my renal blood flow. So that's what I think he wants you to understand. I think he wants you to understand that most of the fluid that enters my kidneys is going to be reabsorbed. And so by the time that the fluid enters into my urine or becomes my urine, it's much, much, much lower than the amount of fluid that enters my kidneys. Okay, so only a small amount of fluid that enters my kidneys actually becomes the urine. Okay, so this is showing you the urine flow. And just to get you guys oriented, if I have a less volume or less urine volume, that's going to lead to lower urine flow. Okay, so let's try to solve this part over here. So the minimal value of very slow urine, okay, think about slow urine, it means low volume. Do you think that's going to be caused by extreme dehydration or extreme water intake? If I have low urine volume or low urine flow, is that dehydration or water intake? Dehydration, good. And then the opposite, if I have higher urine flow, that's caused by extreme, what? High water intake, exactly. It's just intuitive. If you drink more water, you're going to pee more. That's like essentially just what it's saying. Okay, and I believe this is the last slide before I do a little bit of practice questions. This is a pretty cool concept. So if you look at the animals over here, they're seeing how concentrated the tubular fluid gets, right? And what they've noticed is that certain animals, right, have very concentrated tubular fluid. In other words, eventually becomes very concentrated urine compared to some other animals, which have very dilute urine. So beavers have relatively dilute urine, and then desert spring mouse has very concentrated urine or very concentrated tubular flow, right? Like they're showing you over here that the urine of a desert spring mouse is 25 times more concentrated than their blood. Why do you think that is? Why do you think some animals have very concentrated urine and some animals have relatively less concentrated urine? And I wrote a hint over here. Think of the climate. Because he lives in the desert and it's hot, so they don't want to lose water. Exactly. They have very concentrated tubular fluid because they're reabsorbing as much water as they can. That's going to make the tubular fluid or the urine very, very concentrated. That's why the desert spring mouse has very concentrated urine. Good. Okay, any questions so far? I have 3 practice questions after this, and then you guys are done. Any questions, or is it clear? Okay, we're saying all clear. 3 questions. Now let's start with the first one. Which of the following urea transporters transfer its urea from the tubule into the collecting duct cell? Is it A, B, C, or D? I shouldn't have said, I just realized I made a mistake. This shouldn't say collecting duct cell, just keep it as cell. So sorry about that. which of the following urea transporters goes from the tubule into the cell? Okay. Okay, so I see two correct answers. A. The correct answer is A. I'm just going to go back to the slide since I saw two wrong ones. Maybe that was my fault because I phrased it wrong in the beginning. But let's go back over here. Essentially, so remember, oh, it was the collecting duct cells, right? Sorry, I just doubted myself. But I was asking what transporter transports urea from the tubule into the collecting duct cell. So it's UT1. Okay, none of the other ones do that, right? I think somebody might have said UT4, but that's going from the cell into the blood. What I was asking was from the tubule into the cell. Is that clear? Okay, it's clear, good. Okay, which of the following is true about increased vasorectal flow? Oh, I think I actually forgot to explain this concept. If you guys want to answer it, you can, but if not, I'm going to explain it again. So which of the following is true about increased vasorectal flow? A, increased vasorectal flow will increase the interstitial concentration, or B, increased vasorectal flow will decrease the interstitial concentration. I'll explain it again regardless, but I just want you guys to give it a try. See if you can apply your knowledge. Okay, people are saying B, and that is correct. So I'm going to explain why that is. I'll go back to my diagram over here. So it's pretty much just applying the knowledge that you already know, but think about, so we talked about how the interstitium over here, the solutes over here are going to be reabsorbed back into my vasa recta, right? And so if you just think about it logically, if I have more blood flowing through my vasa recta, or the blood is flowing faster, that's going to allow for more reabsorption from the interstitium. And because more solutes are leaving the interstitium, that's going to lead to a lower concentration of solutes inside the interstitium. So it's just an application of your knowledge. So that's why increased vasorectal flow will decrease the interstitial concentration. Okay, last question, which of the following is true? The ascending loop of Henle is only permeable to water. B, tubular fluid that enters the distal convoluted tubule is hypertonic. C, the concentration of solutes decreases down the loop of Henle. D, fluid entering the descending loop of Henle is isotonic with plasma. So I'll just give you a minute. D, good job, guys. And then let's see if you guys, just to make this harder. First of all these three questions are hard. I made them intentionally hard because I want to make sure that even the small details in this lecture, everybody understands or everybody can get correct, right? But if you were able to answer all of my questions before, you're doing really well. So don't worry. These ones are specifically hard. Okay, so try to fix these ones with me. So the ascending loop of Henle is only permeable to water. How would I fix this? What is the correct way to say this? Ascending loop of Henle is only permeable to salts. Good. And then the descending limb is only permeable to water. That's the other way you can fix this. Okay, tubular fluid that enters the distal convoluted tubule is hypertonic. How can I fix this? Hypo, right? The fluid that enters the distal convoluted tubule is hypotonic because it's after the ascending limb and it's already given up all its solutes. So it's hypotonic. The concentration of solutes decreases down the loop of Henle. How can I fix this? Increases. Exactly. The concentration of solutes increases down the loop of Henle. That's it. That's it for my lecture. If you guys have any questions or anything, you can always email me or text me. Okay, no problem. I hope you guys benefited.